Kenji. Okay, so please.
Okay, yeah, thank you, Nakai-san, for organizing this seminar. And I'm impressed that more than 100 people are joining online. Yeah, so yes, so the title of my talk today is what is the digital brain of the Brain Minds 2.0, right? So as Nakai-san said, the Brain Minds project has been Japan's major brain science project, started in 2014, and it was a 10-year project, and then officially concluded yesterday, March 31st. And then this project focused on the marmoset brain, and there was another sister project called Brain Minds Beyond, which focused on more human brain science. And then last year, the Ministry of Education, Science, and the Culture called MEXT made an announcement that for the new brain science project called the Brain and Neuroscience Integration Program, which we now call Brain Minds 2.0. Right? So, and this was announced together with the budget request for this fiscal year last year. And then they said like 5 major topics, like, sorry for being only in Japanese here, but the technology development and the research infrastructure and research on neuropsychiatric disorders, mostly in like a cellular, like a molecular level, and also higher level cognitive function and psychiatric disorders, and the C's for like therapies. And then the important feature is that in the center of this program is so-called the digital brain. And then they call for like a proposal of a core organization for which RIKEN Center for Brain Science was just awarded. And now they are calling for the individual research projects. So this is the website for the core organization. So the RIKEN, the proposal was accepted under the title of Understanding Brain Functions and Disease Pathophysiology through the development and application of brain data integration platform. So in addition to RIKEN, so several partner organizations, Todai, Kyodai, and the QST, and NCNP, NIPS, ATR, and OIST are kind of included in this core organization. And then the important part of this web page is that this lists the like, program supervisor and the program officer for the project. So actually, these people are people who evaluate the proposals. So my talk today is the intention of giving people a better understanding what we are hoping to do in a core organization around the digital brain. But if you submit the proposal, the people who evaluate your proposal is these people. So we are not involved in the evaluation. So this is just information sharing. Okay, so this is the call for research proposals. The deadline is April 10th, 2 p.m. in the afternoon. So there are different branches, and then area 4 is the digital brain construction. And there are 2 types of proposals, team type, which is a 30 million yen per year over 6 years. Another is the solo type, that is a 12 million yen per year over next 3 years. So, and if you go to this site, you can download the call, but probably it is not easy to understand the entire scope of this program and also what the core organization is going to do, especially regarding a digital brain. So that's why I decided to give a kind of informal talk today. So this is the slide we used for the hearing for the center organization. And this is its like English translation. So, and then we have to define what is a digital brain, right? So we defined it as an integration of anatomical and physical and behavior data into a mathematical model to reproduce brain dynamics and functions, right? So, and to reproduce the brain function, like perception, motion, cognition, would make an important contribution to the basic neuroscience and brain-inspired AI. And also the predicting the effect of a change to the part of the brain would be helpful for contribution to the understanding of the pathology of the brain and then the creation of diagnosis, therapy, or prevention methods. So, and then this is a 6-year program. So probably it is impossible to build a complete model of the human brain in 6 years. But we try to develop helpful tools toward developing the digital brain. And then after 6 years, we aim to produce open software for constructing such a digital brain and also prepare some online platform so that people can access and then build their own model and then run simulations. And then the target of application can be like understanding of the brain function, for example, reinforcement learning or Bayesian inference, and also for the understanding of the mechanism of different kinds of neuropsychiatric disorders. So the project will produce a lot of data about the structure or anatomy of the brain, and also the brain activities and the behaviors. And then our digital brain is supposed to kind of integrate these data to produce a kind of working model. For that, we have to do things like multi-species data integration and the cross-species data search, and also the data-driven model building. And then the fitting the model to the experimental data, and also the systematic simulation to understand the brain function and the pathology. Right? Okay. So that is the kind of outline. And then I will try to explain what kind of data and the technology are being produced from the core organization side. So, and the major species we are working on is the mouse, marmoset, the macaque, and humans. So we are obtaining data in a macroscopic level, mostly using MRI, or mesoscopic level, for example, using like a tracer injections, and then like a cellular and molecular level, using different imaging methods, right? So, and for example, in cellular molecular level, so the map of gene expression from the marmoset brain is continued to be developed for the marmoset, and also for the neurodegenerative disorders, propagation of altered proteins like alpha-synuclein is also being measured. And also the post-mortem tissue analysis of human brain is performed, and also the accumulation of different molecules in the brain is being measured by the PET using newly developed markers. And also continuing on from the previous project, we have a cohort of neurodegenerative disorder people, and then going to improve the database, including MRI data. And also there's an effort to try to build a different structure of the brain, starting from a mouse to the humans. And for the physiological data, so different kinds of virus vectors are being produced for mice and marmoset. And also the wide field two-photon imaging is also planned for mouse and marmoset. And also the behavior analysis using motion tracking is a quite important component. And also again, the continuing from previous project, different data sets of functional MRI from psychiatric disorder patients are going to be reinforced. And then to support these data gathering, so there are different technologies are being developed, for example, like fluorescent markers and virus vectors, and also the genetically engineered disease model marmoset, or molecules for chemogenetic manipulation, and also the finding out the genetic blood biomarkers, and also the probes for the PET measurement. Right. So, and these are what the central organization is envisioning, but in about 3/4 of the budget is allocated for the individual research proposals. So we expect that under the current call, different kinds of new technologies or different kinds of new data is supplied into this project. And also there are kind of infrastructures for the omics studies, and then two-photon or MRI imaging, and also the motion capture for the behavior, and also for the digital brain, like computing servers located in RIKEN for shared use. And also we can utilize the databases, brain databases constructed in the previous projects. Okay, so that is the kind of outline. Any questions up to this point?
So I am very big question. So, so what is the digital brain is a very important thing, and you are explaining that integrations of that these kind of data. But so but actually I'm already I'm still so wondering that how do you integrate this kind of your own approach? So how do you integrate these data?
Yeah, so for example, the data about like a cell type. So what kind of neurons are in which their brain areas, and also any information about the connectomes. So what kind of connection are there would be the basis for constructing a model. And also the different kinds of activity measurement, like a two-photon or ECG or MRI, would allow us to like calibrate the model, right? So so verify that the model is reproducing right dynamics or right functions. So these are kind of a basic way of combining different kinds of anatomical and physiological data. And probably for the disease modeling, so for example, accumulation of pathogenic molecules over long term, and also the progress of the cognitive function disorders, that kind of data should be used for like a more long-term prediction model.
Mm. Yeah, but so this is generally it's okay. But for example, the neuron cases, there is a very small neuron. We can see the activity from functional imaging, but so we cannot very difficult to get the connectivity from the neuron to the neurons. So for example, the diffusion MRI data is a very sort of rough structures so we can get, and the tissue side cases. So what I was showing that, but this is also very so base. So not the neuron specific cases. So how to deal with the different scale of the neuron and fMRI, and so these kind of how do you integrate?
Yeah, so as I said, probably it is impossible to build a complete large-scale model of the brain using today's technology in the next 6 years. So we have to select the target. For example, rather than going down to a very detailed level, more focus on like a macroscopic level, like a functional model, or on the other stream, so focus on the like a more like a cellular level, how different molecules cause dysfunction in a cellular or synaptic level. So in the early stage, so probably we have to select the target based on the kind of reliable data available.
So my final question is that do you have already specified or making that a concrete goal of that for 3 or 6 years?
Yeah, so I will come back to that point in the later slides.
Mm-hmm.
So in the core organization, we have 6 subtopics. First is like mapping the brain atlas across different species, for example, marmoset and humans, and also the creating a database which allows cross-species search of like a brain structure or molecules and genes and so on. And then in my group at OIST is supposed to produce like a data-driven model building tool, right? So based on the available experimental data or literature data or certain assumption that we compile a neural network model, and then the evaluate the performance with the known neural activities or functions, and then optimize the model structure and the parameters. And then such a model is used for like large-scale systematic simulation using high-performance computing, and then try to reproduce certain brain functions, and then try to understand the neural mechanism behind that, and then simultaneously, so try to understand how such a function can go wrong. So with the change in the circuit or like components like cells and molecules. So and that is the kind of outline. So then the application target. So for the understanding of brain function or brain computation, what I am interested to understand is that the brain's mechanism for like energy efficient, data efficient learning and the meta learning, right? So yeah, so today's AI is very advanced, but still those neural network models simulated on a GPU takes a lot of energy and also takes huge data for high performance. So, for example, many people, including myself, speculate that cerebral cortex implements like a dynamic Bayesian inference, including finding the right latent representations. And also the basal ganglia is involved in reinforcement learning for new behaviors. So how such like a process is implemented in the cortical or basal ganglia circuit would be a very important topic. And then to really understand, reproduce the mechanism of Bayesian inference or reinforcement learning in the brain. So we need information about what kind of neurons are there. So these days, single cell connectome, single cell transcriptome give us detailed information about the composition of cells in the different areas. And also there's going to be some connectomics information, maybe not only from the Japan, but also in the worldwide, US Brain Initiative is producing such data, and we can incorporate data from other projects as well. And also the measurement of neural activities while the animal is performing sensory prediction or reinforcement learning tasks using mice or a marmoset would be very helpful, right? And then we need to develop technologies like systematic model building or model compilation based on like biophysical data or computational constraints. And then also the automatic parameter search to fit the model to the activity or functional data. And then to test such a function, we need not only the brain, but model of the body and the environment to test how the brain works with the environment. Right. So maybe there are many more things to be worked out. And then regarding neuropsychiatric disorders, while I try to build a healthy brain, probably we will find a lot of strange brains. So they may be kind of a model of the psychiatric disorder by themselves. But once we have a decently working brain model, we can start exploring how such a computation can go wrong. So many of the hard diseases is due to unknown interaction of multiple genes or genes and environmental factors. So that is something we should explore using digital brain models. And then by exploration and then systematic search, we should look for some ways for like early diagnosis or therapy or prevention. So and then so building such a model, certain features of like a genetic or structural or activity level of the patient's brain. It can be human patients or like a marmoset neuropsychiatric disease model brain. And also for the neurodegenerative diseases. So spread of the pathogenic molecules are very important factor. So that kind of data would be very important for building a model. And then how such a change can lead to the like cognitive dysfunctions. That is also kind of very important in constraining the model being built. So the many of the technologies are common to that we need for the understanding brain function, and then the especially for the formation of the pathogenic molecules, the like interaction of the molecules in the intercellular or extracellular space may be important, and also the spread in the brain, and also the damages caused by them. And then model exploration for the to find to reproduce different phenotypes would also be very important. Okay, so and there are many different things needed, right? So these are just what I can kind of imagine at this moment. But as we move ahead, we would find other missing data or more technologies that we need to develop or borrow. So, by the end of the 6-year project, we aim to produce a software tool implementing, for example, like a model or the data search using large language models, or like a data integration, and then multi-species transfer, and then model building, and high performance computing, and optimization. And also the GUI or generative AI to support people building models and then performing in silico experiments. So we hope to make these software tools available for people to use. And also some sample implementation of such integration of such tools as online platform is something we aim to deliver. Right, so the platform can access multiple databases, including those produced by the BrainMinds 2.0, and then people can select the data and then produce the model on the cloud, and then run simulation and visualize the result, and then such a constructed model and then the simulation result should be also shared on the online platform. Okay, so that's the kind of things we are now envisioning. This kind of effort calls for contributions from kind of wide communities, like experimental, clinical, computational neuroscience community, and also AI and data science and software engineering and high performance computing. So last year we had like a workshop to promote collaboration between neuroscientists and mathematicians and data scientists. So I hope to keep that kind of event to promote collaborations. And then also the building a digital brain is something we cannot do just in one country. So, for example, international brain initiatives, e-brains in Europe, or like Allen Institute in the US, and many other places producing data and tools for the relevant for the digital brain. And then we would like share data and reuse tools and make those tools interoperable to build a really reliable digital brain through a kind of in the international community. Okay, and also doing this kind of development needs people. Right. So, and then having seminars like this, or even doing training courses or hackathons to get younger researchers and students interested and involved would be very important. And also we hope to have some industry partners who want to utilize such a digital brain, or support computational infrastructure for such a digital brain operations. Okay, so that is the kind of information I prepared for today's talk. Thank you very much for your attention. And then I would like to answer or further discuss any issues. Thank you very much.